Mitochondria, often referred to as the powerhouses of the cell, are central to the biology of aging. These organelles generate the majority of cellular energy in the form of adenosine triphosphate, ADP, while also regulating apoptosis, calcium homeostasis, and oxidative stress. As we age, mitochondrial function typically declines, contributing to fatigue, tissue degeneration, and the onset of age-related diseases such as neurodegeneration, sarcopenia, and cardiovascular disorders. Yet among long-lived individuals, LLIs, particularly centenarians and supercentenarians, mitochondrial health appears to be remarkably preserved. This preservation may be a key reason they maintain physical and cognitive vitality well beyond the expected human lifespan. One of the defining features of aging mitochondria in the general population is the accumulation of damage to mitochondrial DNA, mtDNA, which impairs the organelle's ability to produce energy efficiently. This dysfunction often results in increased production of reactive oxygen species, ROS, creating a feedback loop of oxidative stress that accelerates cellular aging. However, LLIs exhibit lower levels of mitochondrial oxidative stress and maintain higher ADP production relative to their chronological peers. This suggests that their mitochondria are not only less damaged, but also more efficient at managing redox balance, a fundamental requirement for sustained cellular health. Studies have shown that cells from centenarians demonstrate greater mitochondrial respiratory capacity, improved mitochondrial membrane potential, and fewer deletions or mutations in their mtDNA compared to aged controls. These qualities support better energy metabolism, enhanced autophagy, the process by which cells recycle damaged components, and more effective cellular communication. Moreover, LLIs tend to maintain metabolic flexibility, allowing their mitochondria to efficiently switch between energy sources such as glucose and fatty acids, which is critical for adapting to physiological stress. The integrity of the mitochondrial network, how mitochondria divide, fission, and fuse fusion, is also better preserved in the oldest old. A well-balanced cycle of fission and fusion allows mitochondria to repair themselves, distribute energy evenly, and remove defective units through mitophagy. Disruption of this network is commonly observed in aging and neurodegenerative diseases, but LLIs seem to retain a healthier mitochondrial dynamics profile, which may underpin their resilience to cellular damage and inflammation. In addition to functional characteristics, genetic factors, particularly mitochondrial haplogroups, may influence longevity. Haplogroups are inherited lineages defined by specific mtDNA variations, and some have been associated with extended lifespan and reduced disease risk in certain populations. For example, haplogroup J has been linked to increased longevity in Northern and Western European cohorts, possibly due to its association with reduced ROS production and altered mitochondrial coupling efficiency. Similarly, haplogroup D4A, found in high frequency among Japanese centenarians, may confer protection by promoting mitochondrial genome stability and more efficient oxidative phosphorylation. The mechanisms by which these haplogroups influence mitochondrial performance are still being investigated, but several hypotheses exist. One possibility is that they confer subtle alterations in the structure or function of respiratory chain proteins, making energy production more efficient and less damaging to the cell over time. Another is that these haplogroups interact with nuclear genes involved in mitochondrial maintenance, enhancing repair and turnover of damaged components. Interestingly, the offspring of centenarians, who are statistically more likely to reach old age themselves, often exhibit similar mitochondrial advantages, including higher energy production rates, lower ROS levels, and better metabolic profiles. This suggests a heritable component to mitochondrial health, likely reflecting both genetic inheritance and lifelong exposure to favorable environmental and behavioral conditions that support mitochondrial function. Lifestyle factors play a critical role in maintaining mitochondrial health across the lifespan. Physical activity, caloric moderation, consumption of antioxidant-rich foods, and stress management have all been shown to enhance mitochondrial biogenesis, reduce oxidative stress, and improve mitochondrial efficiency. These interventions may mimic, to some extent, the naturally occurring mitochondrial traits observed in LLIs, offering a proactive route to preserving health span. The mitochondria of long-lived individuals serve as a living blueprint for how energy metabolism, oxidative balance, and cellular repair systems can be fine-tuned to support life well beyond 100. Far from being passive bystanders in aging, mitochondria are active regulators of longevity, influencing not only how we feel as we grow older, but also whether we remain resilient in the face of age-related stress. Understanding how mitochondrial function is preserved in LLIs and how we can replicate that preservation represents one of the most promising frontiers in the science of healthy aging.